Hello everyone, welcome back to my channel Exploring Biology with Sandeep. Today I'm here with a topic that is the plasma membrane. As we discussed yesterday in the last video, the components of the cell, the three main components of the cells are the plasma membrane, nucleus and the cytoplasm. So today we will discuss the plasma membrane in this video. So what is the plasma membrane? Plasma membrane is the outermost covering of the cell in the animal cell, but in the plant cell there is one more membrane, that is the cell wall. But behind the cell wall there is also a plasma membrane. That means plasma membrane is a membrane which is present in the both plant and animal cells. So this is the outermost covering. It's a living membrane, thin, delicate and elastic membrane, which is the selectively permeable membrane in nature. Now why this is the selectively permeable membrane? Because this membrane allow only some material to be entered into the cell which is required by the cell. Not all the material can be entered inside the cell. So this is known as the selectively permeable membrane. Now here we are on the structure of the plasma membrane. Basically the plasma membrane is made up from the two material, that is the protein and the lipids. Two scientists, that is the Singer and Nicholson, that suggested the fluid mosaic model, in which model they tell about us the plasma membrane is made up from the two particles or the material, that is the proteins and the lipids. Proteins are the two types, that is the extrinsic proteins and the intrinsic protein. Intrinsic proteins which are present in between the layer plasma membrane within the bilipids or the phospholipids, but extrinsic proteins are the proteins which are present on the periphery of the plasma membrane. Fine. So there is a bilipid layer of a phospholipid layer, that is the round portion in the bilipid layer is known as the hydrophilic head, and the tail one is known as the hydrophobic tail. The function of the plasma membrane is allow to material or allow to exchange the material within and out of the cell. So here is one more diagram which is giving us a more clarification about the plasma membrane. Here the two layers, that is the phospholipid bilayer is here, head and tail portion, and the proteins are present here. Both type of the protein, that is the integral and the internal proteins. Fine. Next, now how the substances are just move inside the cell and out of the cell through the plasma membrane. Basically the plasma membrane works with through the some processes, that is like osmosis and diffusion, through which the substances move in and out of the cell. Fine. So first let us discuss about what is the diffusion now. So first let's discuss about the diffusion. What is the diffusion actually? Diffusion is a process in which the gas particles move from the region of its higher concentration to the region of its lower concentration until it will get a uniform concentration. Fine. So basically we can say that the movement of the gas particles from its higher concentration to its lower concentration, that is known as the diffusion. For example, you can see a amoeba cell in the diagram. Fine. Now what's going on here? If less oxygen will be present inside the cell and more carbon dioxide will be there, then the movement of the oxygen is from surrounding to the cell, and of carbon dioxide is from inside of the to the surroundings. Fine. So this is an example of the diffusion. This is just an ambiguity which is showing us how diffusion works. Next is the osmosis. Now what is the osmosis? Basically the movement of the water molecules from its region of higher concentration to region of its lower concentration is known as the osmosis. In osmosis we can see there are different type of the osmotic solutions. For example, hypotonic solution, isotonic solution and the hypertonic solution. What happens in the hypotonic solution? If we will keep a cell inside a water, fine, then what will be there? If the concentration of the water inside the cell is less and outside is more, then the water will move from the surroundings to the inside of the cell. In this case the cell will swell up. So this will be known as the hypotonic solution. Then in the second case, isotonic solution, this cell will having the same concentration as in the surroundings. So there will be no net movement of the water molecules from the higher to the lower concentration. So the cell will remain the same in size. So this solution will be known as an isotonic solution. Next, we having an hypertonic solution. What will be happen in the case of hypertonic solution? If we will keep a cell inside water, same, the water concentration is outside is very less and more inside the cell, then the water will move from the inside to the surroundings. Fine. That means water will lose the water. In this case, the water will be the cell will be shrink. Fine. If this case happens in the plant cell, then it will be known as the plasmolysis, and it will happen in the animal cell, then this will be known as the cremated. Fine. So one more example we can give to be explain this more osmosis, how it happens and how it works. We can give the examples of resins that we are usually using at our home for the cooking purposes, for the eating purposes. Fine. So if we keep the raisins in the salt and sugar solution, what will be there? This raisins will be shrink due to the less concentration of the water, and due to the more concentration of the solute, the water which will be present inside the raisin will move from inside to the surroundings due to less concentration of water in the surroundings. Fine. Same if we keep the raisins in the pure water, then what will be happen? The raisins will be swell up, because from the pure water there is no solute is there. From the pure water, raisins will get water and it will be swell into the size. Fine. So these both are the examples of the hypertonic and hypotonic solution. When the raisins will swell up, this will be the case of hypertonic solution, and when the raisins will be shrink up, this will be a case of the hypertonic solution. Let's move to the last topic of the plasma membrane, that is the endocytosis. Now what is the endocytosis? Endocytosis is basically is the ingestion of the material. The plasma membrane also work with the endocytosis. Fine. So endocytosis is further that is the phagocytosis, in which the animals or the other organism can eat the solid food material. Fine. Photocytosis, in which the cell can drink the water and any another liquid material. Fine. So we can give the example of the phagocytosis in the amoeba. Like we all know the amoeba having the pseudopodias, and how it intake the food and how it digest and how it remove the waste from the material just with the help of one cell only. Fine. So the solid food material which is near side to the amoeba membrane or the pseudopodias, they engulf that food and start eating this food. Fine. Ingest that it, then after digestion of that particular food will be occur inside their body, and after the digestion, the waste material will be removed from the outer membrane, that is the pseudopodia in the amoeba. Fine. This is an example of the phagocytosis, how the amoeba intake the food. Fine. Now next is the exocytosis. Exo means to say exit, the cell material which is not able to be digested. Fine. That is removed by the cell through the process of the exocytosis, through the cell membrane or the plasma membrane only. And in exocytosis, secretion of hormones, enzymes and transport of the substances will also be there. So this is all about the plasma membrane. So if you have any doubt as in any queries, please let me know, write in the comment section, so that I can able to be read your queries and I will clear it into the next video. I hope you like this video content, and please, if you like, then do share and subscribe my channel. Thank you so much for your wonderful cooperation. Thank you all of you.